In popular usage, CRISPRs are specialized stretches of DNA. The protein Cas9, or CRISPR-associated, is an enzyme that acts like a pair of molecular scissors, capable of cutting strands of DNA. CRISPR technology was adapted from the natural defense mechanisms of bacteria and archaea, the domain of single-celled microorganisms. These organisms use CRISPR-derived RNA in various Cas proteins, including Cas9. CRISPR stands for Cluster of Regularly Interspaced Short Palindromic Repeats.
In order to change a DNA sequence, in other words, edit a genomic sequence, two things are needed, which are a DNA sequence domain and a nuclease, which has the capability of creating a double break in a specific location in the DNA sequence. The CRISPR locus are short repeats of DNA and are usually 28 to 37 base pairs. There is also spacer DNA that is usually 32 to 38 base pairs that is between each repeat and is regular. CRISPR is a genome editing technology that uses the natural DNA cutting enzyme in bacteria called Cas9, CRISPR-associated protein 9, that can target and edit particular genes. The bacteriophage will attach to the surface of the bacterial cell and will inject its genome into the cell. The viral genome will force the cell to produce the viral proteins and change the cell's machinery. The CRISPR system is a kind of memory, or like the adaptive immune system in humans, that can prevent the same bacteriophage from infecting the cell a second time. There is a three-step mechanism. The first step is spacer acquisition. When the bacteriophage infects the cell for the first time, the bacterial cell will chop up and take a piece and insert it into the spacer DNA. Therefore, the spacer DNA is really pieces of viral genomes that have infected the cell previously. Near the CRISPR locus are Cas genes that produce enzymes needed for the CRISPR process and are usually nucleases and helicases. There are two main players, known as Cas1 and Cas2, where both are dimers and they can form a complex. Cas1 has nuclease and integrase activity, and Cas2 has endoribonuclease activity. The second step is CRISPR RNA processing. One of the two strands of DNA is going to be transcribed into mRNA, which is going to be complementary to the DNA template, and we can call this DNA strand the coding strand. There are three types of CRISPR RNA processing. Type 1, the CRISPR repeats loop and the mRNA will be cut by enzymes, so the mRNA will be chopped up into small pieces of RNA, and each piece has a looped CRISPR sequence and the viral genome piece, collectively known as CRISPR RNAs. Type 2, tracer RNAs will be RNA pieces bound to CRISPR sequences on the mRNA and will be chopped up by enzymes and form CRISPR RNAs that contain the tracer RNA, the CRISPR sequence, and the piece of the viral genome. Type 3, the simplest, where an enzyme chops up the mRNA and will produce the CRISPR RNAs that have the CRISPR sequence and the viral genome sequence. The third step is interference. In general, the CRISPR RNAs will be integrated with the Cas protein that forms a complex of the Cas protein with the RNA sequence, and this is the interference.
Here is an example of a real-life application of the CRISPR technique. As many know, Norway is known for its salmon farming, and the salmon genome has been used to discover methods to solve for issues such as bottlenecks, which could both negatively impact the salmon industry and the natural ecology. One example of a bottleneck that the industry would like to avoid is the genetic mixing of escaped farmed salmon with salmon found in wild populations. The genetic integration of farmed salmon with the wild, native salmon population could cause undesired effects such as disease outbreak and nutrient overflowing to the sea, among other issues. Therefore, scientists thought of utilizing the CRISPR technique to render the farmed salmon sterile. The sterile salmon would be produced using the CRISPR-Cas9 method to prevent the formation of germ cells by editing the genome. By producing sterile salmon, this could be the solution to genetic introgression and precocious maturation. This project became known as the Salmo-Sterile Project and was run by a Salmo-Sterile consortium. Before trying to knock out the genes that would be involved in sterility, the researchers needed proof that the CRISPR-Cas9 method could work on Atlantic salmon. So they applied the CRISPR technique to two genes that produce skin pigment, and the result showed that the technique deleted the genes and caused graded levels of phenotypes. This was proof that the CRISPR technique could genetically modify genes in salmon, and that was enough for CRISPR to be used as the main method to produce sterile salmon. Although the sterile salmon showed advantages into solving the industry's bottleneck dilemma, there were ethical and social questions which were posed as a barrier for the salmon industry to adopt this new technique of genetically modifying consumer goods. Much more discussion will be needed to decide whether the salmon industry in Norway will adopt the CRISPR salmon. Nonetheless, the Salmo-Sterile case illustrated science-based evidence that there is a positive application of the CRISPR-Cas9 technique to produce sterile salmon.
As with any new technology, there can be scientific bumps in the road. An imperative objective within CRISPR is a safety concern that it is moving so quickly, and some researchers want to dive into human clinicals right away, despite the fact of CRISPR technology not being fully developed. Understanding the application and how CRISPR can be used to the advantage, we can also see disadvantages associated in the environmental and health-related safety concerns. This technology will affect humankind and our environment, so as this technology continues, it is imperative that safety measures are being implemented. The main concern surrounding CRISPR is that the technology is still in development, and knowledge about manipulating genes that are implemented to the genome remains very limited. Many scientists advise that the technology still needs a lot of work to increase its accuracy and make sure that changes made in one part of the genome do not introduce changes elsewhere, which could have unforeseen consequences. In addition, another critical issue is that once an organism, such as a plant or insect, is modified, they are difficult to distinguish from the wild type, and once released into the environment can endanger and have an imperative impact on the biodiversity. There is still a discussion to determine whether this approach is safe and effective for use in people. In the United States, the Food and Drug Administration closely monitors safety of any investigational drug. Thus, all CRISPR drugs intended to go into people would have to meet the rigorous testing standards. This is particularly important when it comes to the use of technology for applications directed towards human health. Safety technologies need to advance in corresponding with the CRISPR technologies such that it can be used in a responsible and safe manner to eliminate these challenges.
To conclude this video, CRISPR technology is a simple yet powerful tool for editing genomes. It allows researchers to easily alter DNA sequences and modify gene function. Its many potential applications include correcting genetic defects, treating and preventing the spread of diseases. CRISPR technology has also been applied in the food and agriculture industries to engineer probiotic cultures and to vaccinate industrial cultures, for example, yogurt, against diseases. It is also being used in crops to improve yield, drought tolerance, and nutritional properties. There were ways to edit the genomes of some plants and animals before the CRISPR method was unveiled in 2012, but it took years and cost hundreds of thousands of dollars. CRISPR has made it cheap and easy. CRISPR is already widely used for scientific research, and in the not-too-distant future, many of the plants and animals in our farms, gardens, or homes may have been altered with CRISPR. In fact, some people already are eating CRISPR food. CRISPR technology also has the potential to transform medicine, enabling us to not only treat but also prevent many diseases. We may even decide to use it to change the genome of our children. CRISPR is being used for all kinds of other purposes too, from fingerprinting cells and logging what happens inside them to directing evolution and creating gene drives.